Hi, hello.
Good
Um,
afternoon.
yes, welcome and welcome to anyone joining us online. My name is Julia Pollack, and I'm the current artist and curator for the Art of Science program at the Carl R. Woese Institute for Genomic Biology. Our interdisciplinary science institute not only supports an incredible genomic research, but we also work to communicate science research to the community. So I have a guest with me today. Will you please introduce yourself and tell us a little bit about what you do at the Natural History Survey?
Hi. Well, thanks for having me. My name is Chris Taylor, and I am a curator of fishes and crustaceans at the Illinois Natural History Survey. And as curator here, I kind of have two different focuses. One is to use the specimens in those collections for my own research, and that mainly focuses on studying the taxonomy and evolutionary history of aquatic organisms like fishes and crustaceans. The second focus then as curator is managing all aspects of the long-term care and management of over 1.2 million specimens in just those two collections. The Natural History Survey has several other biological collections, a very large insect collection, mollusks, reptiles, and amphibians, but my area is just fishes and crustaceans. So just in those two um, collections, we have over 1.2 million specimens. And of course, managing that, means everything from making sure that the data, location information, for example, that's associated with a specimen, that's proper and correct, and it's served accurately online. We have web portals for scientists and anyone interested to come in and look at our holdings and find out which species were collected where, when they were collected. So, overseeing, that data management side, but then even down to making sure that the ethanol preservative levels are at appropriate condition for all the jars of specimens that we have.
Cool. That it's wild to imagine. Will you tell us a little about just what is featured in the original image that I reached out to you about?
Yeah, so um, that is a specimen of a shorthead red horse that was collected from a river in north central Illinois. Most people aren't familiar with a shorthead red horse as a fish, but it's a member of the sucker family, and I know that kind of seems like a somewhat silly name, but that's kind of the lay name for this group of fishes, and they get that name because they have a very large mouth that's on the bottom of their head, and they generally feed by swimming along the bottom of a river or stream and just kind of vacuuming up or sucking up food items on the bottom. So very, very large lips and a mouth that's on the bottom of the head, and they use that to kind of suck up food items on the bottom. So that's where the sucker name comes from for that group of fishes in Illinois. And this is just one of the 15 or so species of suckers that we have in Illinois.
Awesome. Um, and so the particular image that I was working with is a CT scan, and we're going to talk later with one of our core members that is familiar with the CT scanning.
Okay.
But can you say anything about how imagery influences your research? So it could
Yeah.
be on any
So,
level.
yep. So imagery, if you are in the business of diagnosing and describing new species, which taxonomists and evolutionary biologists do, imagery is incredibly important. It allows us to describe and disseminate information about shape, color, size of any organism. Um, for example, you can describe in writing what a new species or a specific structure looks like, but the old saying, "A picture is worth 1,000 words," is certainly true, and an actual specimen is worth hundreds of thousands of words when you actually touch and feel and measure and count things. So, if you imagine you have you've found an organism the course of your field work, and you need to identify it. You can go to the literature, and when you see an actual image of the structure you're looking at, it's way easier to make a determination of what you're looking at by seeing imagery, be it, colored pencil drawings, color images, or, things like the CT scans. I don't personally use CT scans in the course of my research, but lots of other researchers do. And the beauty of the CT scan is it allows you to see all, or have access to all that information that's below the surface of the skin of the specimen. You can get into bone morphology, bone shape, bone size. You can even look at internal organs in some cases. So it unlocks a whole new set of characters or information that you can't see without dissecting the specimen. So it's extremely helpful for researchers.
Awesome. Thank you so much. Yeah, it was such a joy to work with this image and to hear about what you do. I really appreciated when we were originally talking, you sort of mentioned that people, as sort of above ground people, we have a lot of sense of creatures that live above ground. So like how many birds you can name or think of, and just sort of thinking about aquatic species.
Yep.
And fishes was such an interesting idea to me because we are not directly aquatic. So
Right.
we just don't have a sense of the diversity of fish that are even in central Illinois where we don't
Right.
have an ocean.
Right. Yeah, no, it's hard to go fish watching as compared to bird watching.
Yes.
But we have 214 different species of fish in Illinois, and a lot of folks just don't realize we have that kind of diversity. And that's kind of one of the functions of a natural history collection, is to keep actual specimens that researchers can come and examine anytime. You can use, you can extract genetic data from the specimens that we have preserved. You can look at diet of an item by looking at stomach contents in specimens that we have preserved. So there's a whole array of information that's associated with these things that we take care of.
Yeah. And it was really interesting too for me, like the piece is split up. So obviously the sample is at the top, but at the bottom, it was really cool you mentioned it before, but try to bring in some of the data into the piece as again, sort of a reminder that the way we have a relationship with like water in Illinois might be like, we think about, there's Lake Michigan, and it's our big body of water, but there's all of these waterways that exist all around the state. And just seeing this fish sort of like all over this map was fascinating to me, to remind me, there's water throughout this area.
Yeah. And, when you're driving sometime and you cross over a bridge and you look down and see a river or stream, I mean, just when you think there could be 20 different kinds of fish, up to 30 different kinds of fish just right below you at that moment, at any given time. So it's a whole other world.
It's crazy. I guess one more thing you do this is unprompted, but what if someone were to find a sample, how do they send it to you?
An email. The tough part is preserving it properly.
Got it.
Lots of times we'll get images. Someone's caught a fish, for example, or a crayfish. They said an image of someone that was out walking in Busey Woods over the weekend and saw a crayfish out wandering around. And we'll send a picture. If the picture's good enough, we can take a look at the picture and identify it. But if it's something that's worthwhile, it's a unique record from a unique area, or, for example, an area where a species hasn't been encountered for many years, we try to find a way to get that record or that specimen preserved properly, so we can accession it into the collection.
I didn't mean to imply that people should just send you a lot of fish.
No. no.
But I wanted to say something just about how people should go and take a look at the Natural History Survey's online portal
Yeah.
and really get a feel for the collection. And
Yes.
it's a really cool collection, and I think it's really exciting for people to know that this kind of work is happening here.
Yeah, great. Thank you. I hope the project increases awareness.
Yes.
Looks really cool. I really love the imagery, and especially in the backside where you can see the different types of scans and type of characters you can get from that scan, and some of the gorgeous colors that go with some of those data layers.
Yes, absolutely. Well, thank you so much. I really
Okay.
appreciate it.
Okay, no problem. Good luck. Take
Bye.
care. Bye.